All right, thank you so much, everyone, for joining. I'm going to get started as these last few people trickle in here. But I'm going to get started by introducing our speaker. Thank you to Patrick Ryan. Patrick is a vice president of the observational health data analytics at Janssen Research and Development, where he's leading efforts to develop and apply analysis methods to better understand the real-world effects of medical products. He's an original collaborator in the Observational Health Data Sciences and Informatics OHDSI, a multi-stakeholder interdisciplinary collaborative to create open science solutions that bring out the value of observational health data through large-scale analysis. He served as the principal investigator of the Observational Medical Outcomes Partnership, OMOP, a private-public partnership chaired by the Food and Drug Administration, where he led methodological research to assess the appropriate use of observational healthcare data to identify and evaluate drug safety issues. We're delighted to have Patrick here with us today to present and share some of his latest work with us. Thanks, Patrick. I'll hand it over to you.
All right, thank you, Pooja. Thank you, everybody, for having me. Actually, you guys, Tony, you're more than 6 feet away, so I'm gonna take my mask off just so those online can see me. So thanks for having me, and I'm appreciative of having the chance to step in last minute to change the topic for you guys this week. What I'd like to talk about actually is work that George and I have been doing with other collaborators in the OHDSI community, which is really about how do we go about demonstrating reliability of real-world evidence. And I'm going to show some work that is work we've previously done and are working on a publication, but also some really exciting results that are very much right hot off the press that we're excited to share with you. So disclosure, so I work at Johnson & Johnson. I'm not going to be talking about any J&J products in this presentation. So we're talking about trust in real-world evidence because my assertion is that there's actually a big problem with the status quo of how we generate evidence from observational data. We're supposed to follow a process whereby we try to we write a protocol, which may or may not be reviewed by someone, but then we go through this process of curating data, identifying cohorts that represent our patient population, implementing analyses, and then we try to disseminate evidence. And usually this is through the form of us writing peer-reviewed publications. And that's at that point that our evidence, we ask the question, is our evidence reliable? And that review of that answering that question can be actually biased because you now are looking at the results. And there's lots of reasons why that observational evidence may not be reliable. There are methodologic concerns, like whether or not you've applied biased methods, whether or not your phenotypes have measurement error, whether or not you have fundamental data quality issues in your data. And there are also a series of technical problems, like how do you actually verify that you wrote code correctly, that the logic that you've applied is correct, that your ETL is correct? And given that we're in this process of doing basically trust me science, the challenge that we have is that there's lots of reasons not to trust the science that's being generated from EHR and other observational sources. And so within the OHDSI community, George and I and others have been really thinking hard about how do we actually try to create a different paradigm of generated evidence that can kind of overcome these challenges. So everybody on this call hopefully is aware of OHDSI. We've created the world's largest network of collaborators and data sources for us to try to generate standardized evidence. And so the work that I'm going to be showing is one of our large-scale experiments that we conducted a few years back. To situate this presentation, though, I want to highlight when we think about evidence being reliable, one way to frame that is what evidence is actually usable for purposes of guidelines in making regulatory decisions. So the US Food and Drug Administration has specific guidelines that it provides to the pharmaceutical industry that basically defines what it actually means for evidence to be reliable. And specifically, what does it require for you to take some sort of action like get a medical product approved and provide labeling statements so that you can communicate to providers. And one of the statutes is that you need to create evidence that's consisting of adequate and well-controlled investigations. And a lot of times this has actually been misunderstood to be stating that to get a drug approved, you must run randomized clinical trials. But rather, it's not about that it is a magic of randomization, but it's actually the burden of proof of how do you prove that the evidence that you're generated is truly considered adequate and well controlled. And clearly randomized clinical trials is one mechanism of achieving that, but it's not the only one. Within the statute of guidelines, they define the criteria for what makes adequate and well-controlled investigations. It's that work needs to have a clear statement of objectives, and that's to basically avoid investigator bias. That a study uses some sort of design that permits a valid comparison with a control, that's to avoid selection bias. That methods for selection of subjects provide adequate assurance that they're from the disease or the condition being studied. So that's really a question of measurement error. That the method of assigning patients to treatment and control group minimizes bias and is intended to ensure the comparability of groups. Really, they're talking about the risk of confounding. That adequate measures are taken to minimize bias on the part of the subjects, observers, and analysts of the data. Again, an issue of selection bias. That methods of assessing the subject's response are well-defined and reliable. So this is really a question of outcome misspecification. And then finally, that the analysis of the results of the study are adequate to assess the effects of the drugs, which is really a question of statistical model misspecification. Now, all of these issues have nothing to do with whether or not you're conducting a prospective data collection for a randomized clinical trial or a retrospective analysis of EHR. However, these threats to validity are very real concerns when analyzing electronic health record data. And we as a community have not actually done all we can to actually prove that our evidence is adequate in the face of these threats. So the question becomes, how do we know that we can trust real-world evidence? And one possibility is, given that people do trust randomized clinical trials, maybe we could compare the evidence of real-world data to the evidence from RCTs. Now, if we're going to make comparisons between answers coming from two different sources, that means we're going to be trying to measure concordance. And it turns out that there's a lot of different measures of concordance that one can consider. One could consider these to be statistical distributions, and one could run a z-test to basically compare whether or not two distributions are statistically different. One could also just say, what's the answer of one study and does it fall within the confidence intervals of another? And whether you're using the referent cohort or the referent estimate or the comparator estimate could change your frame of mind there. From a regulatory policy decision-making, oftentimes statistical significance is used as a criteria. So one either thinks about something as either statistically significantly decreased, statistically significantly increased, or not significant. And one could just ask the question, do you agree in terms of your statement of significance? And one can also consider conducting some sort of meta-analysis to understand, is the information content of an additional estimate, does it actually improve your knowledge or not? Regardless of what concordance metric you want, we need to have some sort of measure to basically answer the question, does real-world evidence agree with a randomized clinical trial? And one of the things that we sought out to do is to start to understand, given whatever metric we choose, we need to actually understand a benchmark of how well would we expect evidence to perform. And it turns out, just as a very simple thought experiment, that if I take those 5 different measures of concordance that have been published and people have used in various studies, that if I take examples of studies here shown on the left-hand side, where I just take an estimate like this first one here, if I've got an estimate that's statistically significant decreased for study 1 and statistically significant increase for study 2, it turns out that all these different measures of concordance all would fail agreement because those are clearly different studies. However, for all of these other examples where one could produce estimates, we can actually see that the choice of concordance metric is actually going to impact what you conclude. So if it's the case that there is not universally agreed statistics for how to measure agreement, and the choice of metric we choose is going to produce different results, then it's incumbent upon us when we're thinking about comparing evidence to actually use this range of concordance metrics and to have a really good understanding of what to expect when we apply those metrics. So before I'm going to answer the question of how well does real world evidence compare to randomized clinical trials, I need to first understand, like, what should I expect from these metrics? Well, one way we could do that is to try to understand how well randomized clinical trials actually agree with each other. And to do this, a few years back, we took the clinical guidelines for hypertension, which provided a wide range of recommendations of pharmacotherapy for hypertension. it was based on this idea that there's a grade of evidence that one could determine to make those decisions and recommendations. And a lot of their recommendations specifically came from randomized clinical trial evidence. Specifically, we focused on the question of choice of initial medication. So in hypertension, there are a wide range of alternative drugs that are available in various classes, including thiazide diuretics, ACE inhibitors, ARBs, calcium channel blockers, and the like. And on the basis of randomized clinical trial evidence, the current American College of Cardiology clinical guidelines made arguments for which classes of drugs would be most effective as initial first-line therapy. Turns out that in that guideline, they identified 4 thiazide diuretics, 10 ACE inhibitors, 8 ARBs, 7 calcium channel blockers, representing 29 different drugs in 5 different classes that are all considered recommended first-line therapy. And that was differentiating those from 28 different drugs that were actually highlighted as secondary candidate treatments for their antihypertensive properties. So even after all these great randomized clinical trials were done, we're still basically left with patients having to make a choice. If you're newly diagnosed with hypertension, you basically get to choose amongst one of 29 drugs that are considered to be all equally effective as it relates to the guideline. But nonetheless, there's randomized clinical trial evidence to support some of this assertion that was being made. Specifically, what made this guideline stand out to us was that they conducted a network meta-analysis across the randomized trials so that they could produce effect estimates comparing each class of drugs with each other class of drugs, and provided a reference of which randomized clinical trials contributed to that direct and indirect network meta-analysis. So this plot basically that they provided throughout their guideline basically has all the 5 classes of drugs as the nodes, and then the edges represent the set of clinical trials that were produced or used to then produce a composite effect estimate for some outcome. In this example, I'm showing you the effects of myocardial infarction. So if I just take one of those arrows there, the comparison of ACE inhibitors versus thiazide diuretics, the arrow that they basically showed was that there were 3 clinical trials that provided estimates of this particular edge. The ALL-HAT trial, which compared chlorthalidone versus lisinopril. It was a very large clinical trial that was specifically focused on patients previously treated with stage 1 or 2 hypertension and who had cardiovascular risks. And in that study, they observed no difference between chlorthalidone and lisinopril with an effect estimate of 1.01. The second study was the ANBP2 study, which compared hydrochlorothiazide versus enalapril in a population of elderly Australians. And there they observed an increased risk of myocardial infarction associated with hydrochlorothiazide with a relative risk of 1.47. And then the third study that made up this network meta-analysis was the Phyllis trial, which was a small trial comparing hydrochlorothiazide versus lisinopril. And this study was so small that they couldn't even report an effect estimate. Now, they took the estimates of all these 3 trials, combined them together in a meta-analysis to basically report that there was no difference between ACE inhibitors and thiazide diuretics because they observed a point estimate of 1.2 with a meta-analytic confidence interval from 0.78 to 2. So the question is, before we start talking about randomized clinical trial evidence and how well it matches real-world evidence and how it matches randomized clinical trial evidence, we could start by asking the question, well, how well does randomized clinical trial evidence agree with randomized clinical trial evidence? In this specific example, this conclusion of no difference was based on 3 trials. 2 trials disagree with each other, and 1 fails to even produce an estimate. So to think about this, it's important for us to imagine what would be the desired target trial we would like to implement if we're trying to draw a head-to-head comparison between 2 hypertensive treatments. What we would like to do is randomize patients to one of 2 alternative treatments. Here it's like ACE inhibitors versus thiazide diuretics. We would follow up those patients following randomization, and we may consider different causal contrasts of interest, such as an intent to treat analysis or an on-treatment period while they were exposed. And then we could evaluate the efficacy for some sort of range of outcomes, including various cardiovascular measures. And we could do this using a very typical analysis plan. So what's interesting is all of those randomized clinical trials that are used in the hypertension guideline to specifically inform the decision of initial medication, none of them follow this target trial design. Rather, there are some variant of some question but whereby we need to actually draw some inference from the randomized clinical trial to the question that we actually want to answer. So that actually makes it really challenging. If I go back a step to this example of these 3 studies, we don't know whether the difference between the all-hat trial and the ANBP2 trial, is it because hydrochlorothiazide and chlorthalidone are different? Is it because lisinopril and enalapril are different? Is it because all-hat studied patients who are first or second stage hypertension with a cardiovascular risk factor, whereas ANBP studied elderly with a particularly high systemic blood pressure? It's not actually clear what the actual effect is that is causing this difference. All we know is that there is indeed a difference. So with that in mind, we were able to extract from the clinical guideline 15 randomized clinical trials that tried to replicate the same class comparison. And so what you're looking at here is the list of these, where effectively for a given outcome, exposure class and comparator, we're able to identify trials that could match up. So for example, this first row, for the outcome of MI, there was the study, the UK PDS 39 study, which compared captopril versus atenolol. And we could compare that to the Agarwal clinical trial that compared lisinopril versus atenolol. This would give us a sense of the comparison of ACE inhibitors versus beta blockers, even though the target drug might be a little bit different. Turns out that in all the work and all the billions of dollars that have been spent to study hypertension, there's actually only 15 randomized clinical trials that actually answer the specific same causal contrast between drug classes. And this is actually the results when we actually put the effect estimates from those trials side by side. So you're looking at 15 plots here with the randomized clinical trial estimates showing you side by side. So for example, this box here is that comparison of all-hat versus ANBP. And we can see it depends on which concordance metric we use to determine whether or not we trust this evidence. So if we use a z-test, we would pass and say that these things agree. However, if we use any of the other metrics that I highlighted before, we would say that these 2 studies disagree because all-hat showed a statistically significant increased effect, whereas ANBP showed no difference. Here's an example where most of the metrics would agree. However, statistical significance agreement fails because one is significant and one is not. And here's an example where they also disagree in terms of these clinical trials. Now, overall, we can take these 15 clinical trial comparisons and produce an expected benchmark comparison of how much we would trust randomized trial evidence. And what we learned was that if I use this first one as an example, using a z-test as your concordance metric, we observed that across these 15 comparisons, 87% of the time, randomized clinical trials agreed with other randomized clinical trials. And you can see the associated statistics across these metrics. Now, this provides us a benchmark to say, how well do randomized clinical trials agree with other randomized clinical trials? Now that we have that as a benchmark, we can go back to our original question, which is to say, how well does real world evidence agree with randomized clinical trials? And how well do our numbers going to compare to this frame of reference? So many of you in the department have heard about Legend, which is an effort in OHDSI to conduct large scale evidence generation across a network of databases. We've established this set of guiding principles for how we generate evidence at large scale. And how we actually conduct analyses across a wide host of different questions using current best practice methods for generating observational evidence. Within our Legend hypertension study that we conducted, we used this idea of a target trial design to specifically create a comparative cohort analysis whereby we compared monotherapy of each and every individual ingredient versus monotherapy of every other contrast. We examined the outcomes of both cardiovascular effectiveness as well as a wide host of safety outcomes, and we produced estimates that we've made publicly available. The only difference between the target trial and our observational work is we don't have the benefit of being able to conduct randomization, so we use large-scale propensity score adjustment as our strategy to try to make these groups as similar as possible. Now, I told you before, there are 29 different ingredients that are recommended as initial first-line therapy, and another 28 ingredients that are out there as possible secondary treatments. What we did was basically permute through all of the possible combinations of alternative hypertension drugs, meaning that there were quite literally millions of comparisons of different hypertension treatments that we could potentially evaluate in the Legend experiment. All of these were, all those that had sufficient sample size were calculated and made available. And we did this across 55 different outcomes of interest, covering both cardiovascular effectiveness and a wide array of labeled side effects. So in total, we conducted hundreds of thousands of different analyses to try to provide us some sort of grounding of real-world evidence that we could look at. We conducted best practice methods that many of you have heard George and I talk about in various capacities. What I'll just highlight here is that we were trying to emulate that target trial to answer the specific question in the clinical guideline as best we could. And we conducted this analysis across a wide range of databases in the US, Europe, and the Asia Pacific region, so that we could try to have some sort of semblance of understanding of generalizability of our findings. And in total, we produced over 1.3 million effect estimates that we were able to generate and make publicly available on a website that have now been out there for 4 years now, that are all available for you to explore. But the question isn't, can we generate the evidence? The question is this evidence reliable? And so we have published a wide range of clinical papers. Some of them have been accepted and been used in various capacities, and some of them have been debated for the question about whether or not they're reliable. For example, George published a paper on comparing chlorthalidone versus hydrochlorothiazide, and there were promptly multiple letters to the editor saying, well, it's just observational research. We got to hold off on trusting that because observational research sucks. So what we did in this first round of the results I want to share with you is we compared the real-world evidence we generated in Legend versus the randomized clinical trials for the same exact drug-drug outcome comparison. We were able to identify 31 randomized clinical trials where we could produce a direct comparison with the estimate from the Legend results set. And this is the findings that we had from that. So each one of these, again, is a plot. The red estimate is the randomized clinical trials, and the blue estimate is our Legend estimate. And so you can see examples. For example, here, if I compare the A and BP study versus, which is enalapril versus hydrochlorothiazide for the outcome of stroke, which produced no difference with a relative risk of 1.02. You can see our Legend estimate had strong agreement. Our estimate of enalapril versus hydrochlorothiazide for the outcome of stroke, our relative risk is 0.98. So across all of the metrics, we would say that this is in alignment and agreement. We had other examples where many, but not all of the metrics agreed. So here, for example, the all-hat estimate, lisinopril versus chlorthalidone for the outcome of stroke, had a statistically significant estimate of 1.15. Our Legend result had a non-significant estimate of 1.14. So it kind of depends on whether or not you are a slave to a p equals 0.05 as to whether or not you think that those two studies agree. But that's why it's important to consider multiple different concordance metrics. But we also have examples like the value clinical trial comparing valsartan versus amlodipine, which produced a statistically significant increased risk of myocardial infarction with a relative risk of 1.19, whereas our Legend estimate of the same question produced a relative risk of 0.89. So under all concordance metrics, we would say we disagree. But the question becomes not to look at these individual anecdotes, but overall, across this entire corpus of studies, how well does real-world evidence agree with the randomized clinical trials? And what we observed across our comparisons is that real-world evidence following the Legend principles agrees with randomized clinical trials, as well as randomized clinical trials compare with themselves. That is, namely, that we observed our concordance z-test of 87%, which is the exact same proportion of agreement that was observed when we compared those 15 clinical trials to each other. And consistently across all of these different metrics, we're able to observe that we're basically in the same ballpark. That is to say that if you believe that concordance is an objective that one would want to achieve, that concordance between two trials and concordance between a trial and real-world evidence following the Legend principles gets you about the same ballpark of agreement. Now, one could say that this is useful, but we still don't know theoretically what the estimates of these concordance metrics really should be. So another way to think about benchmarking would be to conduct a simulation whereby what you have fixed is the sample size from the randomized clinical trial and the sample size from the real world evidence. And we could actually say, let's imagine that the effects estimates truly are in agreement. Then statistically, what would we expect to be the concordance metrics if we were doing a perfect replication? That is, we would to just randomly sample from a truly unbiased estimate. And it turns out that if we were to do that simulated perfect replication, that a concordance z-score, as you probably all learned in Stats 101, would produce the true answer 95% of the time because we've set our p-value at 0.05. And you can see the associated other metrics. What's important to see here is that with their z-score, we produced 87%, just like the trials. We're a little bit below what we would consider to be theoretically perfect replication in a truly unbiased state. And when we look across the range of other statistics, we can see we're largely in line. This gives us tremendous encouragement that at least in this retrospective view, when we compare legend results to randomized clinical trial results, that we can be confident in saying that our level of agreement that we're seeing is on par with the fact that we must have adequately addressed a bias because we're seeing results that are consistent with unbiased estimates. Now, this is all retrospective evaluation. You know, we already had the results of the clinical trials in hand. We already we conducted our study, and one could say, you know what? I bet you what you guys did was you just like tweaked the data and you did a whole bunch of p-hacking until you got the answers that you wanted. But we also did something else a couple years ago that was quite fun, and this is the results that are kind of hot off the press that I'm really excited about that George has been leading. So back in 2018, when we showed all of these results, we also highlighted one specific point in the clinical guideline, which is this top bullet, which said that chlorthalidone is preferred on the basis of preferred half-life and proven trial reduction of cardiovascular disease. And specifically, it's referring to preferred as it compares to hydrochlorothiazide, which is another diuretic treatment. And back with the clinical guidelines, this is basically based on a weak physiologic hypothesis and indirect comparison of the clinical trials, because no such direct head-to-head clinical trial existed for this question. And so it turns out that back in 2018, when we were reporting this, there was a clinical trial that was underway called the Diuretic Comparison Project. And specifically, they weren't exactly answering the target trial question. Rather, they were taking people who started hydrochlorothiazide, and then they chose to either switch them to chlorthalidone or maintain their treatment on HCTs. And they were basically going to eventually report out the outcomes to basically determine whether or not chlorthalidone was truly the preferred diuretic of choice. The issue was that back in 2018, when we talked about this, they were estimating to enroll 13,000 patients. The trial had started in 2016, and they weren't going to report out until October 22. Think about what calendar date we are now. So the question we put on the slides back in 2018 was, what can we learn from observational data while we wait 4 years until this randomized clinical trial is completed? So we didn't just wait until today, but rather George led the OHDSI group in publishing a paper in Journal Internal Medicine that compared cardiovascular and safety outcomes of chlorthalidone versus hydrochlorothiazide. And in George's paper, we summarized that across the cardiovascular effects, we observed no difference in myocardial infarction, hospitalization for heart failure, stroke, and composite cardiovascular disease, with calibrated effects estimates that were all very close to 1. And pretty sufficient power for us to be able to comfortably rule out that there was a big difference in cardiovascular effects. What was also particularly interested in George's paper was that not only did we say we don't believe that chlorthalidone has benefits relative to hydrochlorothiazide, but we actually observed a host of increased risks associated with side effects. Very specifically, one that I'll call out, we observed hypokalemia had a relative risk of 2.7, which was highly statistically significant. So not only did we not see benefits of chlorthalidone, but we actually warned of risks that were observed not only in electrolyte imbalance, but also in renal outcomes that was observed through the LEGEND trial. And so we published this, and George was pretty excited that we were going to, hopefully impact clinical guidelines. This followed all of the best practices within OHDSI community and legend studies. So we were able to show that we minimized bias. We were able to show that we saw consistent effect estimates across multiple databases. We were able to perform various sensitivity analyses that showed robustness of our findings. So we felt good about this conclusion. And yet, letters to the editor started popping up, including one that was co-authored by Paul Welton, who is the lead author of the hypertension guideline that I showed before. And in his letter, back to George, they highlighted that chlorthalidone is recommended over hydrochlorothiazide because it is a longer duration of effect, has more extensively documented as effective in randomized trials. Prior meta-analyses and observational comparisons suggest that chlorthalidone is superior to preventing cardiovascular risks. However, to our knowledge, there is no published randomized trials comparing these treatments, but such a study, that diuretic comparison project, is ongoing with expected results in 2023. They then also continue to say moderately strong prior evidence suggests the superiority of chlorthalidone, and there is substantial likelihood of residual confounding that accounts for the lack of observed difference in George's study. And for this reason, it is imperative that we await the more definitive VA trial results in 2023 before changing clinical practice guidelines on diuretic choice. So here we've got an observational estimate, got no randomized trial. And one thing we could do is we could wait until all trials are done to have answers, or we could try to learn from what we could learn in absence of trials. Fortunately, 2 weeks ago, this clinical trial has now reported out. So now we can see what happens when we prospectively validate our real world evidence by seeing how does George's guess at the clinical trial estimate compare to what the trial has now shown at the American College of Cardiology 2 weeks ago. And the results, there you go, November 5th, 2 weeks ago. The results of just top line results were actually presented at the American College of Cardiology. And what they reported out from their clinical trial, here are their principal findings. The primary outcome of major adverse cardiovascular events for chlorthalidone versus hydrochlorothiazide, hazard ratio is 1.04 with a confidence interval from 0.94 to 1.16. Their secondary outcomes, no difference in hospitalization for MI, no difference in hospitalization for stroke, no difference in hospitalization for heart failure, but they observed a statistically significant increased risk associated with hypokalemia. So their interpretation of their trial is that the trial shows no differences in cardiovascular outcomes. They do some exploratory analyses to suggest there may be subpopulations, but they do observe that hypokalemia was more common with chlorthalidone. So just to take George's estimates and put them side by side with the clinical trial estimate, we can compare the legend results that we presented in 2018, published in JAMA Internal Medicine 2020, versus the clinical trial estimates that were reported 2 weeks ago. And just like the trial says there's no difference in cardiovascular events, myocardial infarction, stroke, and heart failure, those estimates are directly in line with what George presented years previously. And that initial signal that we warned about an electrolyte imbalance has now been verified in the clinical trial. So I wanted to go through that because I think what we're now seeing is an opportunity to prove the reliability of real world evidence. And what we've done with the legend work now is not only retrospectively validating by comparing to clinical trials that are already done, but now with this example, being able to prospectively validate the reliability of real world evidence by predicting what the trial is going to show, waiting till the trial is done, and then showing that our results are consistent. Now, it sure would be nice if I didn't just have one anecdote of those. It would be nice if, just like I had 31 retrospective studies, that I could have 31 prospective studies. But our problem isn't on the end of the real world evidence. The problem is that there are not 31 head-to-head randomized clinical trials going on for hypertension right now. In fact, there's none. Now that this study is reported out, there are effectively no large-scale head-to-head comparative clinical trials in this space. And so if we consider that we want to use randomized clinical trial evidence as our gold standard to benchmark real-world evidence against, we're going to have a real challenge because we are actually particularly limited in the amount of randomized clinical trial evidence we have. But another frontier that we have been advocating in the OHDSI community, and we think is a future to look forward to, is not to wait till we have evidence to try to verify that we believe it's true, but instead to build a system whereby reliable evidence can actually be proven at every step of the way. What George and I have been advocating now in the community is that we should be thinking about a series of steps whereby we can first prove that our data is fit for use. Secondly, prove that our phenotypes have sufficiently low measurement error that they can be trusted. Prove that our analysis designs are free of confounding and other sources of bias. And only after we prove those steps a priori do we provide evidence that's made accessible by passing all of these objective diagnostics. And we think that this can be a very different paradigm from our current evidence generation process, whereby we do trust me science, throw out an estimate, and then wait for the reviewers to tell us that our observational data is crap. And instead be able to actually have a proof that our observational evidence is trustworthy, independent of the effect estimate, so that if you can trust the method and the system that's used for generating evidence, then you should have greater stock in the evidence that's produced. And one of the nice side effects of this is if we have a systematic process for generating evidence, that not only can we generate evidence with higher quality, but we can also generate evidence with much greater efficiency. So the current paradigm of largely is reactive and bespoke. Somebody comes up with a hypothesis, they design a study, they generate evidence. If we create a network of data sources, such as what we have in the OHDSI community, that makes it possible for us to think about doing analyses across multiple databases, which is tremendously valuable. But if we want to be responsive in generating evidence to meet the requirements of, for example, clinical guidelines, we need to be able to have standardized analyses that we can apply that actually have already gone through a rigorous process. And if we can generate evidence using standardized analyses, we can actually be prepared to generate evidence at real time. For example, having evidence that's available at the point of care, like Anna had previously demonstrated as part of her dissertation. But even more idealistic, if we can generate evidence that's reliable and can produce it at scale, we should actually be able to anticipate the needs of those that need evidence and push evidence, not just filling evidence gaps in the literature, but actually filling evidence gaps in the hands of clinicians and patients that are making good decisions. What it takes to do this is just standardization. That's why our informatics field really needs to be focusing all of the steps of standardizing data, analytics, and our dissemination paradigms. But the opportunity that's in front of us is that instead of observational evidence currently being a belabored process that takes months or years to conduct, we could actually totally transform this framework whereby we could have evidence in near real time to actually support real health decisions. So I think a really exciting opportunity of following these kind of standardized processes. Not only can we prove that the evidence is better, but we can also prove that we can generate the evidence in a more systematic and proactive way. So just to conclude, and I'll leave 5 minutes for questions before we close it down for Tony's proposal defense. I'm really excited about the results that we have in the Legend validation work, because it's now, for the first time, taking one clinical domain, has been able to show retrospectively and prospectively that real-world evidence can be reliable. What this took was, though, for us to actually have a prospective systematic approach to generating evidence, and the rigor of having a community working together. It required us working through the data and the tools and the best practices all together, as opposed to our current paradigm of generating evidence where everybody's kind of going off and doing their own thing. That said, now that we've demonstrated this both technical feasibility and scientific integrity of the results, I think it's kind of incumbent on us to say, like, how can we now go apply that to other disease areas and other areas of work, which is a great opportunity for anybody within our department who wants to join the fold. There's a lot of open work we need to do to actually like scale this out even further. But I think what I'm most excited about is I think it demonstrates the value and the impact that we could have if we actually work together on this space. So with that, I will stop and happy to take any questions from those in the room or those virtually online here.
Wonderful. Thank you so much, Patrick. So I will be monitoring the chat. Please raise your hand if you want to ask a question online, and then in person, we're gonna use our dynamic problem solving skills here to facilitate any questions in person.
I've got one question here in the room from Chenhua.
Go ahead.
What do you kind of give some prediction about the future? Do you think that what time will replace randomized control trials?
Good question. So Chenhua's question for those on the line was, can I give a prediction about the future about what the role of real world evidence will be and whether it will replace randomized clinical trials? So I think that I think the goal that I would like to see is that when there is not a randomized clinical trial available, we consider that real world evidence could be an opportunity to fill that evidence gap and provide sufficient evidence to make decisions, rather than policymakers saying, let's wait till the clinical trial is done. The reality is, in almost all disease areas, the amount of randomized clinical trial head-to-head studies is extraordinarily sparse. So 90+% of all questions we want answers to don't have answers and won't have answers if we wait for RCTs. I don't think the aspiration should be that we entirely replace randomized clinical trials, in part because many questions that we want to answer, that the outcomes that we want, aren't something that can be reliably captured in electronic health record data on the offset, particularly things that are not just hard outcomes to be able to measure. But right now, our current paradigm is, like in the clinical guidelines, they either go with randomized clinical trial evidence or expert opinion. Even the systematic reviews of most guidelines just completely omit real world evidence. There is a reason for that omission, which is that historically real world evidence hasn't been trustworthy. And so the work that we're trying to do with the OHDSI community is like, let's build the trust so that at least it could be part of the fold. And I think if we could get to that point, what we're going to see is, sure, trust your randomized clinical trial for the 5% of questions that it bothers to answer. And then in the absence of that, let's use real world evidence to fill the majority of the space where there is no answers.
Great. We have a question online from Will.
Oh, sorry.
Yeah. I guess you sort of just answered at the end there, but I guess like you were talking about the value of having people within OHDSI sort of work together and to incorporate this, standardized evidence practices. But how would you start getting other organizations or researchers that are totally separate, like on board, and how long would that take for it to have sort of an impact within the research community?
Yeah, it's a good question, Will. So did everybody in the room hear that? Yeah, okay. So I guess actually George and I were just brainstorming earlier today about this specifically. How do we actually expand the impact of the work we're doing? So I think the first thing is we need to continue to demonstrate that indeed real world evidence following these practices is reliable. And so we need more examples of what we just shown. Across the OHDSI community, we now have thousands of collaborators and the world's largest data network. But the reality of it is, even our Legend study only involved like 10 databases. So we report that we have 400 databases converted to the OMOP common data model. Only 10 of them actually reported out results in this study. So we have a long way to go to actually enable our entire community to generate evidence at scale. That said, the OHDSI community is still a small microcosm of the overall research ecosystem. And I think what we need to do is just have more demonstrated cases where we can actually generate evidence and push it out. And hopefully having these impacts like the community has had in revising guidelines, in supporting our regulatory authorities, the more impact we can have, the more credibility and the more people we can bring into the fold.
And then I saw Kayla, you raise your hand. Do you still have a question?
Yeah, well, and I think this kind of speaks to both of the previous two questions. Kind of when you threw up all those papers and said that the reaction was kind of, well, these are observational studies. It seems like there's kind of a twofold approach of bringing these into practice, which is proving that these results are valid and rigorous, but also just kind of getting buy-in of a paradigm shift. And so I'm just curious, since you kind of like straddle these different worlds, how you see that kind of playing in, of actually convincing people that maybe RCTs aren't as valuable as the gold standard as they've always been.
Yeah, thanks, Kayla. So, I, so I said, what George and I spend most of our time scientifically thinking about isn't about undermining trials, but it is about raising the profile of real world evidence. And one of the sensitivities there is that we're not advocating that any old use of an electronic health record will generate reliable evidence, because actually we've been able to show consistently that like probably 90% of the observational studies that have been published are crap. And so we are not advocating that just like any old use of data is appropriate. But what we've been able to demonstrate is like principled use following specific agreed practices a priori can be credible. And the question is, how do we actually get that message out there in an appropriate way? Of which I would assert very strongly, like George and I don't have a solution to that. If we did, we'd be probably happier than we are right now. But I think we're making progress. You know, this clinical trial that just reported out, you know, we're trying to figure out the effective way to message this, not only because George's paper also highlights other safety issues for patients making that treatment choice that probably matter, but also from the methodologic perspective, to our knowledge, this is the first ever demonstration of retrospective and prospective validation of real world evidence. And so a lot of people have talked about like, wouldn't it be nice if someone could do this, but we just did it. So now the question is, how does this hopefully start to maybe at least change minds, or at least open up some conversations with people who otherwise would be kind of all in on randomized clinical trials only.
I'm not sure if there are other questions in the room, but we have one more online from Chen Yang.
Yeah, thank you. My question is, Professor, do you think real-world evidence research can be sufficient enough to fill the gaps towards RCTs? That's my question.
Yeah, absolutely. So what I see right now is one of our most acute problems in healthcare is that most decisions are made in complete absence of any evidence. And so if we can prove that we can generate evidence reliably, I think real world evidence can fill a huge amount of gaps. The one area that I'm specifically excited about getting anybody in our department who's willing to listen to me to think about is the area of adverse event detection, pharmacovigilance. Because I think this is an opportunity for our entire department to engage on filling a gigantic gap that really real world evidence is best positioned to solve, because randomized clinical trials are never powered to identify and detect rare side effects, but they really matter to lots of patients all around the world. And so I think that's a huge area of opportunity. But even when it comes to comparative effectiveness, the thing that drives me mad is that I would of course want randomized clinical trials if I had them, but the vast majority of randomized clinical trials are placebo controlled or not providing a direct comparison. And indirect comparisons of randomized clinical trials are the equivalent of really, really bad observational studies. And so much like we saw in this guideline, in the absence of trials, using indirect comparisons can lead you to bad conclusions. I think real world evidence can be used to.
Thank you.
All right. Tony's now looking anxious. So I'm gonna call it adjourn this, Pooja, if that's okay.
Perfect.
So
I
that we
was gonna
can
do the
turn
same
it over
thing.
to.
Great.
All right, great. All right. Thank you, everybody.
Thanks, everyone. Have a good holiday.